Hello and welcome to BioCode. In this video, you will see how we can utilize Sequence IO module within BioPython. Seqio module stands for sequence input and output. Basically, it allows us to read sequence files, various sequence files that can be FASTA, GenBank, cluster files, or files, or other sequence alignment files as well. So it allows us to take those files as input so that we can read those files, so we can convert those files, those sequences that are present within a particular file into sequence records, which we saw in the previous video series. So what really happens in sequence IO module is that it allows us to take sequences as input from the user or from the file system or whatever, and it also allows us to write sequence files. For example, if you have created your own sequence record throughout the computer, throughout the processing or the execution of your computer program that you created in Python, and you would like to now save that sequence or save that analysis that you produced, that represents some kind of sequence, and you would like to save it as a sequence file, then you need sequence IO or Seqio module of BioPython that will just allow you to write the sequence in a particular file, which we will study in the next videos. But in this video, you will learn how we can utilize sequence IO module to read a particular file that can be FASTA, that can be GenBank, and you will also learn the difference between parsing and reading, which basically confuses a lot of people. So we will demonstrate you the reason and the actual difference between parsing and reading. Basically, the parsing video will be separated from this one, but you will learn the difference in this video. And sequence IO basically takes two arguments as well. First, the name of the file or the path of the file. Absolute path. It could be an absolute path. It could be an relative path. If you don't know what that means, you need to watch the previous videos about file reading. And the second argument is the file type. For example, if you are trying to read a particular file, you should actually know that either it is a FASTA format, either it is a GenBank format, or some other kind of file format. So let's get started. First of all, we need to import the sequence IO module from BioPython package, and then there are various different. These are just comments that I created for you guys, and then what really matters is that we need to actually read a particular file, right? First of all, let me show you that what file are we going to read, and let's just open it up in VS Code. And okay, so these are the two files that we are going to read. This is this GenBank file of a particular gene, particular mRNA, and it has accession number of this, and this is basically a GenBank file. We are going to read this file as well using BioPython sequence IO module, and we are also going to read this file, which is a FASTA file of this particular sequence. It has an ID, a description of the sequence itself, and the sequence itself. So we are going to read these two files. In the next video, we will parse these sequence, the multiple sequences. Okay, so let's get started. First of all, we need to import a particular module, which is sequence IO. After we have imported this particular module, we need to pass in two parameters to read function of sequence IO module. Read function is basically takes two argument. It takes the name. It could be relative path, it could be absolute path of the file, or it could be the name of the file, like this. Then you need to pass in the format. For example, as I said, we will be reading this my file, then it should be in a FASTA format, right? As we saw in our demonstration, that it is indeed my file.fasta is indeed a FASTA file. So we are actually telling the read function that it is a FASTA file. You need to interpret it as FASTA file. So then we are storing that particular that particular command or that particular statement in a different variable, which is get faster record. Basically what really happens is that once you have read a particular file using sequence IO, it gets converted into a sequence record. So we need to store that sequence record in a particular object or particular variable, so that we can utilize that variable to get the results. For example, we need ID, for example we need sequence, for example we need the description, features, or something like that. So in this demonstration I'm just going to convert it into a particular directory sequence record, and then I'm just printing out the record. But in the next demonstration we will actually retrieve the IDs as well. So same goes for the genbank file. We are actually telling that since it ends with .gb extension, as you can see over here it ends with .gb, and indeed it is a genbank file. So we are actually passing the parameters as sequence.gb, then we are actually passing the variable name to be genbank, the parameter name of the file format to be genbank, and then we are also storing it into a get genbank record variable, and then we are printing it out. So let's do one thing, and let's results from my file are, and then we can do this. We just need to pass in the ID property, and then we also need the description. We can also utilize the sequence. Same goes for the genbank record, and we also need the ID, we need the name, description, and we need the sequence, and we also need the features. Basically this means that we are actually utilizing genbank, so we have the ability to actually extract the features, annotations. Let's make annotations, and it will just extract that information. Let's copy this one as well. Okay, so let's run our code and we will see the results. There we go. Okay, so let's go to the top. First of all, it says that results from my file are, the ID goes over here, the description, then the sequence itself, then the results from sequence.gb are ID, the description, and the sequence. So both of these results are same, because these are the same files, just different formats. One is a genbank format and the other is FASTA format. And it also shows that it does not have any annotations, which is quite literally impossible for the genbank file. Let's make it feature so we can extract some separate information as compared to the FASTA format. And after the running, okay, I think so there's some issue. It looks like there are no features for this particular genbank file. So that was all. In the next videos, this is basically a simple